Hello.
Hi, how are you?
Well, I'm pretty good. I'm still doing graphs, but I see the end.
Oh, good. That's good.
Yeah. I have to do stress strain graphs as well as the distance force ones, the force distance. I don't know.
Okay, yeah.
So hopefully this week I need to hand the whole thing in. So
Oh, okay. Well, that's good. Good luck.
thanks.
Yeah.
I'll probably shred something about it. So anyway.
Yeah. There's Morgan. Hello, Morgan.
I hope I don't have any problems again this week, but I will turn off my video after a bit because I don't know, bandwidth or something.
Oh, yeah. Okay. So, yeah, welcome to the meeting. So I'll get started. In, I think, 2 weeks, I can't remember what the exact date is. I think it's the 17th. We're going to have the annual meeting for Devo or for OpenWorm. And that's where we usually get together and talk about all the different sub projects. And so I have been working on the DevoWorm 2024 update. And so it's actually up on the YouTube channel right now if you go there and look. And so we have the DevoWorm update, and I kind of go over the 10th anniversary of DevoWorm, kind of talk about that a little bit. I have a separate video on that on the YouTube channel. Then I also talk about our Google Summer of Code contributions, talk a little bit about Pocky's project, talk about the lectures we did this year, talk about the papers we did this year. So we did the molecular basis of differentiation wave activity and embryogenesis. Going through that, this paper on hypergraphs, and then this is Mahul's work on spatiotemporal decomposition of the C. elegans embryo.
After PETA was attacked by an animal.
So it should be up on the YouTube channel. I'm going to talk to the OpenWorm Foundation about this in a couple weeks. So they're always happy to see our work and what we've been able to do in the past year. So actually, I'm going to start with this article from Asimov Press. So this is a really good outlet. This is Nico McCarty, who I think runs Asimov Press. And this is an essay on the levers for biological progress. So you have this. This is just an article on sort of how AI is intersecting with the idea of, kind of making progress in biology. Talking about, kind of comparing it to the Human Genome Project, and kind of considering how, maybe if you apply AI to biology, there are going to be a number of shortcomings in that area. So this article starts talking about Dario Amodei from Anthropic. He's the CEO, and he recently published this essay called "Machines of Loving Grace," which is a vision for how AI could radically transform neuroscience, economics, diplomacy, and the meaning of work. So this is a pretty ambitious essay. Amodei also imagines ways AI could accelerate biological research and yield miraculous cures in the 21st century, everything from the prevention and treatment of nearly all infectious and inherited diseases to the elimination of most cancers. So on Saturday, we talked about this in depth with respect to the Human Genome Project, how, the Human Genome Project kind of promised the same thing like 25 years ago. And of course, it wasn't probably a fair set of promises from the standpoint of what could be accomplished on a time scale that everyone at the time would be alive to see. But, we're kind of repeating that with AI. So just to frame that in a framework that, we can kind of appreciate. biology is probably the area where scientific progress has the greatest potential to directly and unambiguously improve the quality of human life, Amode writes. My basic prediction is that enabled biology and medicine will allow us to compress the progress that human biologists would have achieved over the next 50 to 100 years into 5 to 10 years. So this is like a tenfold increase in the speed of discovery or of progress, however you want to define it. So, this is something that, it's pretty ambitious, just to say the least. And so this is, of course, then in this article, they're going to talk about maybe how there are bottlenecks in this process of discovery that probably won't yield the promised acceleration of progress. So it's an inspiring vision, but you have to think about the bottlenecks and the roadmap ways to solve them. So, in other words, there are bottlenecks to making progress. In the Human Genome Project, there were bottlenecks in terms of, bioinformatics, in terms of technical barriers, and those had to be overcome at the same time that that technology was being implemented. So, this at Asimov Press, they published different types of roadmaps. So they had an article on where's the synthetic blood, which was kind of like talking about how easy or hard it is to get synthetic blood and how to grow it in the lab, or to have other types of roadmaps on different topics in biology. So the sort of the sort of the assumption here is that the powerful AI should be smarter than a Nobel Prize winner. But even if you had that kind of super intelligence, could you overcome these bottlenecks? So, we have a lot of bottlenecks in biological progress, but those are biophysical rather than computational. So they talk about that here. And, when I talk about biophysical bottlenecks, I mean they're bottlenecks to understanding different processes. So, and this holds true for, say, like imaging technologies or sequencing technologies. We have a way to collect data now. We have a better way to collect data, but we still don't understand the process that we're working with. And as a consequence, you've seen this with respect to next gen sequencing. We've generated massive amounts of data, but it kind of sits on a shelf because we can't interpret it properly. And so that's probably not going to speed us up. Uh, Even if we have a super intelligence, we might be able to find, say, relationships, but it's hard to actually verify them and then incorporate it into actual progress. So this is, some of these bottlenecks are really going to be hard to overcome, maybe with different technologies that aren't AI, but we'll see. So, again, it's kind of hard to know where solutions will emerge, but, we don't want to overpromise and underdeliver. So that's why researchers hoping to accelerate biology at large should strive to build platform tools that can apply in multiple contexts. So Adam Marblestone has written an article on his Substack about building these platform tools that are versatile rather than narrow solutions to short-term problems. And so this is another thing where you can develop a technology and apply it, but having a platform is better. So this is this Adam Marblestone article, if I can get access to it. Okay. This is called Biotechnological Knowledge Hacking. This is from the MIT Biomaker Workshop in 2016. Adam Marblestone has been associated with OpenWorm in the past, so he's one of Stephen Larson's colleagues, or he knows him pretty well, I guess. So this is they have this sort of connection to MIT. There are other people who were kind of involved with this, such as Ed Boyden and George Church. So this is kind of a lengthy talk. I'm not going to go through it, but basically it talks about how bioengineering is not like normal engineering. You can't necessarily rely on analytical calculations. The underlying systems are poorly characterized with respect to being able to engineer it. So we can engineer, say, an automobile or a bike, but it's hard to engineer a biological system in the same way. We have high failure rates. So In engineering, we try to have a safety factor. We try to lower the failure rate. But in biology, you really can't do that as well. There's a widely distributed knowledge base, so it's hard to know what we know. So, we have to go into the literature and just find, the answers that are spread widely across that literature. So that's kind of what they're getting at with this. So, we can use AI and it can advance knowledge, but it's one of these things we have to manage. It's not just something you can throw at the problem and immediately get a return on your investment. So the balance between AI advances on the one hand and wet lab innovations on the other is also a bit like the chicken and egg problem. Yes, AI will accelerate biological progress, but first we must make it easier and faster to run experiments while creating better methods to study biology in a more holistic and less reductionist way. Solving the latter challenges will, oddly enough, require both machine learning and wet lab innovations. So biophysical bottlenecks are basically, bottlenecks that we can characterize through this quest to build a virtual cell. So there are a bunch of groups that are trying to do this, trying to build working models that can accurately simulate living organisms. And by a virtual cell, it's just simulating that one cell, a eukaryotic cell, but maybe even a prokaryotic cell. A virtual cell would enable researchers to perform biology research on computers rather than actual organisms, and removing challenges that arise from the working in the world of atoms. So we can't necessarily build a virtual cell just through simulation. We need to understand much more about how cells work, and that takes us back to the realm of experimentation and wet lab research. So you can't necessarily predict the outcome of even simple biological questions, such as if I take this gene from an Arabidopsis plant and insert it into E. coli, will the cell express the gene and make it a functional protein? No one really knows this without trying it out in the laboratory. And even if we do get like a meta-analysis of studies, we still have to kind of we don't know what parts we need to abstract away from that system to make the model work in silico. So there are a lot of challenges to this kind of approach. So in order for 50 to 100 years of biological progress to be condensed into 5 to 10 years of work, we will need to get much better at running experiments quickly and also collecting higher quality data sets. This essay focuses on how we might do both, specifically for the cell. Although my focus in this essay is narrow, I don't discuss bottlenecks in clinical trials, human disease, or animal testing. I hope others will take on these challenges in similar essays. So that's that article. I know that sounds like something that Morgan could speak to a little bit, perhaps Susan, because she's doing simulations of, or doing kind of engineering of cells in a roundabout way. So any thoughts on that?
I could certainly use some better data
Yeah.
for my simulations. Like it's missing, like the elasticity of the actin filaments, for instance, that are bundled. The bundled actin filaments will have other molecules included in that. And okay, so what's the elasticity of this? Nobody knows. That's my conclusion. They've done actin, just straight actin. But actin bundles, like there's several different molecules involved in different types of actin bundles. Like, okay, where's the data? Doesn't exist. So that makes my life difficult.
Yeah.
And that gets me into what I'm doing now, something called K factor. So it's the actual area of the filament times its elasticity divided by the rods. It's the same thing going on in the denominator, and you get a K factor, and that will change tensegrity's elasticity, well, how it moves, etc. So it's stress strain curve.
Right.
So it would be good to know what actually is, even if I just had one of them, I would like the K factor of the different types of cells, and then I could do this research easier, but there's no data.
Right.
Yeah. So what is the diameter of the linked actin in a cell that makes these strings that I'm trying to use in my tensegrity?
Right.
What are you talking about? What are you talking about? Yeah, it's something like that.
Yeah. I mean, certainly you can sort of use stand-ins for a lot of these things and model them, but then the question is how predictive are they, or how successfully can you predict something in the system? So if you build
yeah. For instance, a lot of cells are sensitive to the substrate they're on. So how a cell is attached to a substrate is important. And it's also important what characteristics the substrate has. Very important. But I'm just starting to get at modeling that. And of course, my lovely program is won't do everything that I want it to do. Cells seem to be attached in points on the surface of the substrate, and you make a mesh on the bottom of my pseudo cell, and I have nodes, and then you try to attach the nodes to the substrate, and it doesn't work. So somehow this is not a proper thing to do if you were building a building, which is what I'm up against. I'm trying to use software that builds buildings and machinery parts.
okay.
Yeah. But the two should mesh. They need to come together somehow in order to study this.
Right.
You have to put proper engineering into it, proper physics and then engineering into your model, or people just aren't going to believe you. They're going to say, oh, well, it's just a theory. It's just a theory. It's not engineering.
Oh, yeah.
Engineering has to work in the real world.
Right.
Yeah. So, yeah, I'm trying to explain cells in engineering terms, giving me a headache.
Yeah. Yeah. I think a lot of approaches to bioengineering are kind of like that. I mean, if you're talking about, even if you're talking about like a gene circuit, they usually use these toy problems that are pretty simple. And, you can do very simple things with them, but then when you try to go beyond that, it's a little bit difficult. So, there's this tendency to, and this is true in biology too, or, in computational biology, there's this tendency to cling to the easier systems or easier applications. You can do like a proof of concept, but then does it go beyond that? And that's kind of the problem as well.
Well, biology is complicated.
Yeah. Right. Why don't we move on then? Actually, this is a paper Susan might enjoy. This is forecasting avalanches and branched actomyosin networks with network science and machine learning. So we're kind of following up on this discussion about biological engineering and using different tools to sort of accelerate that process. And so this is from the Journal of Physical Chemistry B. And so this is, uh, I'm not familiar with these authors, but they're going to tell us about forecasting avalanches in these actomyosin networks. So this is a graphical abstract here. We have a physical network. We transform that into a mathematical graph. And we try to predict these actomyosin avalanches, which, before reading the abstract, I'm going to assume is some large-scale reconfiguration of the physical network. And you're going to see a large-scale reconfiguration in the corresponding mathematical graph. So you can see that in the graph. You have the structure here, and then it transitions to the structure here. So there's something analogous to a first-order phase transition in that graph. And so that's what they're trying to predict. And, in these actomyosin networks, which remember are these physical networks of actomyosin. So the abstract reads, we explored the dynamic and structural effects of actin-related proteins 2/3, which is ARP2/3 as the shorthand, on actomyosin networks using mechanochemical simulations of active matter networks. And so they were doing these mechanochemical simulations of these networks. I guess active matter means that they have modeled all of the aspects of the actomyosin and put them in these kind of at these interaction points. And we're looking at the interactions, and so you can abstract that to a mathematical graph. On the nanoscale, the ARP2/3 complex alters the topology of actomyosin by nucleating a daughter filament at an angle with respect to a mother filament. At a subcellular scale, they orchestrate the formation of the branched actomyosin network, which is here, the physical network. Using a coarse-grained approach, we sought to understand how an actomyosin network temporally and spatially reorganizes itself by varying the concentration of ARP2/3 complexes. So this is where you get this spatial reorganization of the network, and you get this concentration of ARP2.3 complex or 2/3 complexes. So this is a bit like adding in like something like, it's kind of like the transition between ice and water, how water transitions between a solid and a liquid. So you get this sort of, you're adding in, instead of adding in temperature, you're adding in a concentration of these complexes. And you end up with this phase transition. It's again like a jamming transition as well, where you have a bunch of particles that kind of go through a pipe. And if you get to a certain density, the particles cease to flow and it becomes a solid. And so the flow is restricted, but it's a sudden sort of process. It doesn't happen gradually. And so that's what they're trying to do. They're trying to get these spatial reorganizations by varying the concentration of these complexes. Driven by motor dynamics, the network stalls at a high concentration of ARP2/3. This is where we get this kind of transition. It contracts at a low ARP2/3 concentration. So you get this, remember this network, of course, is responding to forces and stresses and strains. And so it has to have some sort of like movement. And so when you have a high concentration of these molecules, you have the stalling of the network. Whereas when you have this low ARP2/3 concentration, you get contraction. And so these are two sort of extreme states of the network. At an intermediate ARP2/3 concentration, however, the actomyosin network is formed by loosely connected clusters that may collapse suddenly when driven by motors. So you get these kind of clusters here in this mathematical graph on the left. And I guess you can see them here where you have these clusters corresponding in the network. And so these loosely connected clusters kind of sit there in this sort of metastable state. and they collapse suddenly when driven by motors. So when you actually drive the network functionally, they can collapse suddenly. This physical phenomena is called an avalanche. So this is where we get this rapid shift in the topology of the network, largely due to the marginal instability inherent to the morphology of a branched actomyosin network when the ARP2/3 complex is present. So that means that you have these sort of clusters that form at this intermediate concentration of ARP2/3. These uh, clusters are metastable, yet they're fragile. And if you drive them by some sort of motor or some sort of set of stresses and strains, you can collapse that structure and you can get these avalanches or these massive reconfigurations of the network. So you see this here. So that's what they mean by an avalanche. It's this large scale reconfiguration of the network that can happen suddenly and due to some sort of perturbation on the network. And remember, if we have a really high concentration of ARP2/3, the network ceases to be flexible and it just sits there in sort of a mass. And if we have very low concentrations, we end up with this sort of different state where the network contracts and it's also non-functional. Okay. While embracing the data science approaches, we unveiled the higher order patterns in the branched actomyosin networks and discovered a sudden change in the social network topology of actomyosin. So they're talking about, I don't know, I guess they're referring to sort of the way that this resembles a social network. And what they mean by that is that social networks, of course, have been well studied in terms of network properties, mainly like small world networks, where you have the structure that's focused on key hubs or highly connected nodes, and then a lot of nodes that are not as highly connected. So you get this hierarchical structure. You get the small worldness that results in a very short distance across the graph because you're going from the edge, one edge of the graph to a hub to the other edge of the graph pretty quickly. And that has consequences for the structure of the network. So that's what they're getting at with the social network topology of actomyosin. And so what they're referring to here in terms of the actomyosin network and its dynamics is that there's a new type of avalanche in addition to the two types of avalanches associated with a sudden change in the size or shape of the whole actomyosin network, as shown in a previous investigation. So they've been studying this for a while. They have a previous paper where they show that there's a sudden change in the size and shape of the whole actomyosin network, which you see here. You can imagine that this network undergoes some stress or strain or some perturbation, something that's driving it, and it can change very quickly the shape and the size of the network. But you also have this social network topology, which is this hierarchical structure that you get that's a sudden change that the network can transition to. Our new finding promotes the importance of using network theory and machine learning models to forecast avalanches in actomyosin networks. So this is where they're actually trying to forecast these avalanches instead of just observing them. So you can observe them by, perturbing the network, by driving the network and letting it sort of move. And you can see these changes in the actomyosin network. So, but you can also then predict maybe where they might occur. So you could actually, now that you know kind of how these parameters are, you can actually try to make predictions. The mechanisms of ARP2/3 complexes in shaping the architecture of branched actomyosin networks obtained in this paper will help us better understand the emergent reorganization of the topology in dense actomyosin networks that are difficult to detect in experiments. So that's what they're getting at. They're actually able to do these simulations, get these graphs, look at the topology, look at the behavior, and perhaps predict some of these avalanches. And of course, these avalanches would be useful in terms of understanding quick changes in the sort of the composition of um, one of these actomyosin networks in the um, in the or in the membrane of the cell. So they talk a little bit about muscle cells and how actin filaments and myosins are organized into a striped sarcomere. And in non-muscle cells, actomyosin networks tend to be isotropic, especially at the edge of cells, such as the actin cortex. So there are different, depending on the cell type, they're located in different places and they have different functions. They're basically contractile, but you have in muscle cells, you have a very different mode of action than you have in the actin cortex of a cell membrane. And of course, they also have different properties in terms of how they're organized. We hypothesize that the nanostructure of the actomyosin network dictates the structure and dynamics of the entire system. So they talk about the ARP2/3 complex. They talked about coupling this with myosin motors and how this controls contractile tension generation in a cell. Now, different computational models have been used to explore actomyosin contractility, and that's references 7 and 8. But few of these computational models explore the effect of ARP2/3 complex on the dynamics of the system. So people have done these, they built computational models just looking at the contractility of the actomyosin themselves, but they've not really looked at the effects of the ARP2/3 complex on the dynamics of the system. So, you want to look at how this network changes over time. Despite extensive experimental and computational studies, and that's references 9 through 12, only recently has the computational work from our group shown that the presence of the ARP2/3 complex causes sudden collapse dynamics of marginally stable actin-myosin networks. And these are, of course, termed avalanches, as we talked about in the abstract. The avalanche possibly underscores the phenomena of a cytoquake. That's reference 14. A drastic structural change in the actomyosin network within a short period of time. So the cytoquake is in reference 14. The biophysical importance of this phenomena is appreciated since it is deeply related to the structural rearrangement of the cytoskeleton. However, how this phenomena connects to the nanoarchitecture of the branched actomyosin network, orchestrated by the ARP2/3 complexes, remain unclear. So they look at the impact of ARP2/3 concentration in modulating avalanches in these branched networks using coarse grain simulations. They use a software package, Median, which simulates the organization of actin filaments with mechanochemical feedback from actin-binding proteins. In our simulations, even though we only changed the concentration of ARP2/3 complexes, while the turnover rates remain the same in the chemical reactions, the patterns of the temporally evolving networks were incredibly complex. Driven by a high concentration of motors, several new global features or orders emerged from a locally well-connected network. And so they show this figure 1. This is the Median workflow. This is their simulation environment. Median consists of 4 main steps in its mechanochemical loop, as described in this figure.
The mechanical model, is it showing things in tension?
Okay.
Yeah, well, I would like to see it show the system in tension.
Okay.
Because cells are in tension.
Yeah.
Basically all the time.
Right.
Occasionally not, but And if they're in tension, of course, then they're tensegrity, and sometimes the tensegrity will all of a sudden change shape.
Right.
So it's kind of a fit between sort of a avalanche or a catch bond or a change of shape. I don't know, there's some sort of might be some sort of a fit there. I know my tensegrity models have some what they call soft modes where all of a sudden it collapses, and I've got graphs to prove that a couple of my models have that sort of behavior. It'll all of a sudden just there's a sharp change in the stress strain graph. So it's all kind of interesting.
Yeah.
And I ran across a paper that where they made a tensegrity, and then they said it works really well for the first part of it, but then it does this. And I'm going, well, you built a tensegrity. What do you expect? And I can maybe send you the paper.
Yeah.
They tried, but it's not good enough. So I wasn't going to include it in my thesis references, even though it's or maybe I should, just to say, well, it's a tensegrity, so you can observe that this happens to it partway along its behavior, stress strain curve behavior. As you found out, just because I think you might be interested.
Yeah.
I'll send it to you.
Okay.
Or maybe I'll look for it right now too.
Yeah. Yeah. So getting back to the paper, we have this MEDIAN workflow. MEDIAN stands for Mechanochemical Dynamics of Active Networks. And this was developed by the Popoyan group, and they have citations here. So this is basically to include the mechanochemical feedback of the active networks. And so this is kind of, I don't know if they show. Okay, well, that's not what I want. Okay, so this is the Actomyosin dynamics here. This is the workflow of Median, so it consists of these 4 steps. The first step is a chemical model where you have an initiation of the chemical model where the system evolves the Actomyosin network with stochastic chemical reaction diffusion. So we have this chemical reaction diffusion model that's instantiated. Then we update the mechanochemical configurations in 2, and that's where the chemical reactions deform the network locally, followed by the formation of a new mechanical configuration. So we have this chemical model, we have this update of the mechanochemical configurations. Imagine that this is going through a loop that, we instantiate the chemical model at every iteration, then we update the mechanical configurations. Then 3 is a mechanical model where we initiate the mechanical model, and we first initiated the chemical model in 1, and now we initiate the mechanical model in 3. So this is where the total energy of the new mechanical configuration is minimized with the conjugate gradient method by reaching a new equilibrium. And then 4 is update chemical reaction rates, where the chemical reaction rates are mechanically updated at the new equilibrium state. So we cycle through these from 1 to 4 and then back through 1 to 4 and back through 1 to 4. So we're always initiating a new chemical model or a novel chemical model at every iteration. We update those mechanical configurations, then we update the mechanical model. We then update the chemical reaction rates and then go back to the chemical model. So that's how they kind of run their simulation. They talk about simulation parameters. They talk more about this method. And then this is sort of the output of the Median simulation, which are these networks where you have these actomyosin filaments that have contact points where they have interactions And that's how you can take this physical network and turn it into a mathematical network. So these are typical snapshots of median simulations for the unbranched Actomyosin networks without ARP2/3 complexes. And okay, so that's A, this one here on the left. This doesn't have the ARP2/3 complexes. This is just an unbranched Actomyosin network where there's no actual branching at the contact points. So you just see it's like a collection of Actomyosin filaments kind of um, in this box. And then in B we have these branched Actomyosin networks with ARP2/3 complexes, which allow it to form branches, and you have this network with the ARP2/3 complex. So if you have too many of them, remember we said that it would freeze up, and if we have too few, it would collapse or shrink. And so I guess A would be the case where there are too few, B would be the case where it's just right. In both snapshots, a red cylinder represents an F-actin filament. So these red cylinders here are F-actin. A black bead represents a plus end of an F-actin filament. A white bead represents, and I guess you can see the white beads in there, represents the minus end of an F-actin filament. A blue cylinder, which are these blue lines, represent an ensemble of NMIIA motors that consist of 15 to 30 motor heads. A green cylinder represents an alpha-actinin crosslinker. You can see the green ones here and in here. And a yellow bead represents an ARP2/3 complex. So we have yellow beads throughout. You can see that. So that's where we have this model. So this is, what it looks like in the output of the simulation. We have reaction rates that they've kind of derived here. You have reaction rates for actin filaments, linkers, motors, branching, and then destruction. You have other parameters in here. And then the data analysis that they do. So they look at, they draw from network theory to analyze these networks. And they, kind of build a mathematical graph out of this. And so they convert Actomyosin networks into mathematical graphs. The confirmation of the mathematical graph from the physical Actomyosin network and the calculation of network theory order parameters were performed with the Python package network X. So they take these simulation outputs and they turn them into mathematical graphs. So they create a standard graph and then they map everything onto that. They use network X, which is a standard approach. So they talk about the graphs here, where this is, they kind of show the graphs, sort of an introduction to graph properties, which isn't really helpful here. So then they also have order parameters for these graphs. So these graphs are complex systems, and one of the things you can do is use order parameters to sort of understand the structure of the network. So yeah, so they actually do a little bit of work on assortativity, meaning that they want to see, how these networks connect together. So they do this, they establish this criterion for assortativity, and then they use machine learning models to forecast avalanches. These data analytics for machine learning models. So they do some analytics here and they get some results in these graphs here. So they look at the time course of these branch networks with low, medium, and high brancher concentrations. And so I guess low is A, medium is B, and high is C. So low is A here, B is medium, C is high. Okay, so that's what we have there. Then we have this graph here or this figure here where we have the branching networks on the left and the mathematical networks on the right. So this is where we have graph networks of physical actomyosin networks at several ARP2/3 concentrations at 500 seconds. So this is where A, C, and E show snapshots of the simulations at low, medium, and high ARP2/3 concentrations. So A is here, C is here, and E is here. So these are the physical networks. This is a low concentration of ARP23. C is a medium concentration, and E is a high concentration. You can see this ratio here. XB to A is 0.02, 0.0 or 0.02 or 0.002, 0.02, and 0.2. And then your BD and F are the graph representations of those actomyosin networks. So basically you're taking this physical network that's branching. In this case, you don't really see a lot of branches. In this case, you see more branches. And then in E, the network is very highly structured and branching. And so you can see the consequences in the mathematical network. So for the ones where you see very little branching, you have very few edges between nodes. You have these hubs that are sparsely connected, and you have some nodes that aren't connected at all. And that makes sense because if you look at the physical network or the physical branching, it's not very clear that there's a lot of physical branching here. In C, we have more branching, and we have this corresponding network where we have these hubs, we have edges or peripheral nodes, and the edges are more, there are very few things that aren't connected. There are a lot more edges, and there's sort of this clustering that you start to see take shape, where you get clusters of nodes that are connected. And so, like this green cluster here, this blue cluster here, and then this component around the edge that's connected. And so this is indicative of this greater branching and more ARP2/3. Then finally in this F, you see the sort of the translation of E as having a lot of branching, profuse branching, if you will. And you get this corresponding relationship in the network where you get these clusters, this highly structured mathematical network of different clusters, and almost like patches on a quilt. So you see that here. So basically, as you increase the branching, you get more order in the network. And that's really kind of, I think, the insight here is that they're looking at the amount of order that this brings to the network. They're looking at the connectivity, and then they can measure, they can look at these order parameters. They can look at the different uh, graph statistics, and they can look at how the graph is structured, how it's connected, and how it behaves over time. Okay. So I don't think there's anything else worth talking about here. It's just they have a little bit more work on assortativity. And so this is something where you get displacements of filaments, where you get connections that change between filaments. And that's what they talk about at assortativity. So there's this third type of avalanche, which is assortativity. I assume this is what they were talking about when they talked about the social network properties. Because in social networks, aside from the small world behavior, you get this assortativity where there's a sort of like attaches to like. So there's a way of attaching nodes through edges to other nodes. And so this attachment has to happen by some process. Sometimes it's random. Sometimes it's what we call assortative, which means that like attaches to like, or there's some attachment rule that results in some systematic behavior in the network. And so this assortativity is the third classification of avalanche. And so they talk about that in this figure here. So basically, when there's an increase in assortativity in the network, node A and node B now form a center-like cluster during the avalanche. The example simulation is a high motor, low linker, and medium brancher concentration. So the A and B are here. So you have in the first case here, you have node A, and all of these nodes are sort of I guess, connecting to it. In C, you have A, where all the nodes are connecting to each other instead of A. In F, you have where A is connected, everything's connecting to A but not B. And then in the last example, you get B connecting to the same nodes as A. So this is just kind of showing how these assortativity rules change. Okay. So let's go down to the references, because I thought there were a couple of really interesting references in here. So we're looking for reference 7. And reference 7, so they talked about 7 through 12 being sort of indicative of the different types of simulation that are being used currently. So 7 is collective Langevin dynamics of flexible cytoskeletal fibers. That's from the New Journal of Physics. There's also a versatile framework for simulating the dynamic mechanical structure of cytoskeletal networks. It's from Biophysical Journal. Number 9 is molecular transport modulates the adaptive response of branched actin networks to an external force. That's Journal of Physical Chemistry B. And then Popoian, I guess, which is the method that they used. Hough and Popoian, mechanochemical feedbacks regulate the actin mesh growth and lamellopoidal protrusions. That's Biophysics Journal. And then Lan and Popoian, the stochastic dynamics of filopodial growth, and that's Biophysics Journal. And then 12 is Carlson, the effect of branching on the critical concentration and average filament length of actin. So these are all sort of simulation related studies, except for this last one, which talks about the effect of branching on critical concentration. So this is in Biophysics Journal. Then if we go back up to 5, actually, or well, there's 5 and there's 6. So 6 is this paper by Papalazaru and Macheski. The cell pushes back. The ARP2/3 complex is a key orchestrator of cellular responses to environmental forces. So this talks about this ARP2/3 complex and its role in responding to environmental forces. So that's kind of the context for, what the forcing mechanisms are in these networks. So you can build the network and you can simulate it, but unless you know why it's doing what it's doing, it doesn't really mean much. So that's an interesting reference. Number 5 is this reference by Malik Garbi et al. And this is scaling behavior in steady state contracting actomyosin networks. So this is important. This is a Nature Physics paper from 2019. This is important to understanding these different modes of behavior and how they change with a growing network. So this is where we want to know something about the steady state and the scaling behavior of the network. Okay, so then the other one I wanted to get to here was number 14. So actually, there's another article on the role of ARP2/3 complex in shaping the dynamics of structures of branched actomyosin networks. That's number 13. That's from PNAS. So that's another paper on the ARP2.3 or the 2/3 complex and shaping sort of the dynamics of the network. Now, number 14 was about these cytoquake events. So the cytoquake events are these things that are sort of these cell-based or these structural avalanches. And so this is Alcanar et al. Non-equilibrium cytoquake dynamics in cytoskeletal remodeling and stabilization. This is from Soft Matter. So this describes kind of the cytoquake phenomenon. So I don't know, this is again, these avalanches, I don't know. I'm not going to go to the paper right now, but that might be something to follow up on. Okay, so that's good. And then there's some other references in here which are nice to look at. Let's see. There's a paper by Poimanos and Popoian stochastic ratcheting on a funneled energy landscape is necessary for highly efficient contractility of actomyosin force dipoles. So this is physical review X. This is talking about stochastic ratcheting, which I'm not familiar with, but I assume that there's some sort of action on the structure and thinking about the energy landscape and how that manages uh, the efficient contractility of actomyosin. Okay, so I think that's all for that paper. It was an interesting set of things, a lot of things going on in that paper, but basically they're simulating actomyosin networks. They're looking at uh, these avalanches, and they're trying to predict them using machine learning from their output of their simulations.
They need to add more chemicals.
Yeah.
Their simulations, so you can see like the bundling of actin.
Right.
Because that's the branching of actin with ARP2/3 complex, but there's bundling, and that's a different chemicals. So I'd like to see that.
Yeah.
Of course.
Yeah. Well, I thought it was interesting from the standpoint of modeling sort of the nonlinear dynamics of the structure. Now, one could argue that you need to have more things in there because you're just kind of working with a network, and maybe have environmental forces. So you're just kind of modeling the network and saying, we can simulate the forces on the network and they have this effect. But if you had like a cell membrane or a muscle in real life and you had some force on it, like maybe another cell, or, it was pushing against some, like other cell that was migrating past it, some actual real world context where the forces are coming in. I mean, the forces are going to be maybe a little bit more, especially when you're dealing with the dynamics, they might be a little bit different than what they're presenting in the simulations. So getting these sort of isolated modes down, I think, is important, but you also have to have this, observe behaviors that are sort of naturalistic and that they're actually happening. They're actually things that are biological in nature, things happening in the context of the cell.
I think it's scaling up that's scaling up, like adding forces over a whole cell would be scaling up.
Right.
From the molecular levels. So, but I'm very interested in these branches in us because that's where my rods and strings for my tensegrity come from. That's what they are in real life. They're not just a rod; they're a branch network that goes through the cell.
Yeah.
So I need to explain that actually probably in my intro just a bit. So maybe I can use some of their diagrams and say, see, there's a network in the cell.
Yeah, that'd be good.
Yeah.
The other thing I wonder about with this is that they're describing branching dynamics, but when they take it and they move it to mathematical network, they're treating it as sort of connection. I don't know if it's a directional connection or not. It may very well be, but there's a difference between sort of like a interaction between two nodes that's non-directional and one that's directional, and it can have play a role in sort of the dynamics of the network, the mathematical network, not the actin network. So I think maybe they should have maybe used a little bit more sophisticated network theory, or but I don't know how I didn't really dig deeply into what they were doing there. They just had some demonstrations of like an ordered network, but it didn't appear that there was a lot of like that there was an explicit order in the mathematical networks. But
they have.
Oh, go ahead.
Sorry, there's a paper or two about brush bottle networks for cells and how they work. rather than just the straight actin. The ones that have branches like a brush or a bottle on them are behave differently than just this straight up actin. So the last diagram showing all the little branches reminds me of that paper where they said, oh, there's a brush bottle network of actin in the cells. So there's some other papers about some of this branching. And there's also branching that's kind of at 60 degrees. And then if it's being pushed on from the top or something, it becomes a 90-degree angle.
Yeah.
So it holds better. It holds up more force. That's on the cell membrane. I have some, maybe I should show you, I have some papers in a PowerPoint, but you probably don't want to see that today since it is over 100 slides long.
Okay.
Yeah. I don't know, something to do after I've handed in a thesis.
Right, yeah. Gotta take it one step at a time.
Look at that.
Well, I'll have to look into these brush bottle networks. I'd not heard of that before, so.
Okay.
Yeah.
I'll try to send you a couple of papers here. I've been thinking of sending you papers, and then I forget because I'm trying to do a whole bunch of other things and get this done. And
yeah, that's the most important thing. Yeah.
Yeah, just keep focused.
Yeah.
Because I can certainly get sidetracked. So I've been trying not to.
Right.
Yeah.
Okay. Well, any other comments, Dick or Morgan? Do you have anything before we go?
Great stuff. Yeah. Hard to track all the details when I'm kind of multitasking here,
Right,
but yeah, I really like it. There was a nice intro to something from earlier. There was a nice intro video from I forget what the acronym CEMB is, but it's like mechanobiology, I think, is the MBs. Yeah, just talking about a number of just interesting concepts that are covered in mechanobiology and just made me think of they were talking about kind of natural structures that displayed strain dependent properties. So they were calling like viscoelastic and things like that.
Right.
But just again, like that interesting intersection of engineering and biology.
Yeah.
I'll try and drop links later when I got the kids to school.
Okay. Yeah, that sounds good. Yeah, a lot of these networks, they're physical networks, but they also exhibit different types of properties when forces are applied. So they're not like static. They have they deform in different ways and they recover. So how do you represent that as a mathematical network? That's a little bit tricky. So yeah, it's definitely
For me, I'm just have to do it statically. Like it's at this place at this point, and this is the model of it when it's stressed like this. And it's going to be stressed like this a second later because there's a force on it. So it changes. Anyway,
right.
I don't know how to get that into a movie. I suppose maybe one could. I don't know.
Yeah. All right. Well, thanks for attending. Good luck, Susan, on your thesis this week.
Thank you. Yeah. just down to the final few things.
Right.
Yeah, I mostly have it written. Yeah. So, yeah, I should go do that.
Yeah. Great. All right. Morgan?
Just yeah, good job, good job, Susan.
Oh, I'll just wait till it's finished. And they say it's good, and then I'll just go, yeah, enough of this. Then I can do what I want with it.
Yeah. All right. Thanks. Talk to you next week.
Good.
Bye.